Hello everyone. Today we will be solving Biology Paper 1, October-November 2018, Variant 2. Okay, so let's look at the first question. What is the diameter of a typical prokaryote such as Streptococcus? Options A, 7.5 into 10 to the power of 1 nanometer, which stands out to be 750 nanometer. Now it is a good idea to write every one of them in the same unit so that when deciding about their length it's easier. So option B, 7.5 into 10 to the power of 2 nanometer, and therefore, sorry, my bad, the first one was 75 nanometer, the second one is 750 nanometer, the third one, which is 7.5 into 10 to the power of 0 micrometer, would stand out to be 7500 nanometer, and the last one would be 75,000 nanometer. Okay, so the diameter of a typical prokaryote is 7.5 into 10 to the power of 2 nanometer, or 750 nanometer. So option B. A typical plant tissue is first observed under microscope using red light with a wavelength of 650 nanometer. The same specimen is then observed under the same condition but using green light with a wavelength of 510 nanometer. What happens to the magnification and resolution when using green light compared to red light? Now you see, magnification is not going to change because changing wavelength of particular light would not change the magnification. However, wavelength, since we are using now a shorter wavelength, so the resolution will increase. So we will go for option D. It remains the same and the resolution increases. Which structure are present in Vibrio cholera cell? Okay, a Vibrio cholera cell. A Vibrio cholera cell is a bacterium. So since it is a bacteria, of course it will have a circular DNA. It will not have a ATS ribosome, neither would it have a mitochondria. So I would go for option A. Okay, so let's move on to the next question. Question number 4. The removal of a plant cell wall by enzyme digestion leaves an intact membrane-bound structure called a protoplast. Which statement explains why protoplasts are easily damaged? So as you can see, already the cell wall has been removed. Cell wall is removed. So which statement explains why the protoplasts are easily damaged? All right. Option A, mitochondria stops producing ATP to maintain membrane integrity. Well, mitochondria doesn't have to do anything with membrane integrity. Net movement of water is no longer limited to turgor. Could be. Then secretory vesicles are unable to fuse with the cell surface membrane. Nothing. All right. The shape of the cell structure is changed by the loss of cell shape. Okay. So here we have nothing. So net movement of water is no longer limited to turgor. Turgor was maintained by the cell wall. Since the cell wall is no longer present, so the turgor pressure could not be maintained and the cell will just simply burst. Which statement are correct for number 5? Which statements are correct for a green plant? Number 1, ATP is produced by mitochondria. Okay, ATP is produced by chloroplast. Well, both are correct. ATP forms part of DNA. Now, ATP doesn't form part of DNA. Okay, even though adenosine is part of DNA, but then ATP doesn't form part of DNA. So I'll go for option B. Okay, number 6. Which process occur in eukaryotes and prokaryotes? Number 1, hydrolysis. Yeah, can occur. Mitosis. Mitosis will occur in eukaryotes and will not occur in prokaryotes, so I'll leave it across. Transcription will occur in both, translation will occur in both, so I'll go for option 1, 3, 4. C. Question number 7. A sample of a solution was tested for reducing sugar and the result was negative. Okay, solution tested for reducing sugar, result negative. Another sample of the same solution was then tested for non-reducing sugar and the result was positive. Which step in the test for non-reducing sugar breaks the glycosidic bond? Adding Benedict's reagent? Nope. Adding sodium hydroxide? We will add it to neutralize. Boiling with hydrochloric acid? I think that is the correct option. Heating to ATC? Well, heating doesn't break glycosidic bond, so in this case we would go for option C, boiling it with hydrochloric acid. Hydrochloric acid helps it to hydrolyze by adding the water. Now next, question number 8. Which row correctly identifies 3 polysaccharides? Okay, I mean polysaccharides with their uses. Alpha glucose, straight chain. Straight chain would be amylose and amylose. We wouldn't go for glycogen, so option C and D is already wrong. Alpha glucose with branching chain, we would go for both amylopectin and glycogen. Beta glucose with straight chain, well, beta glucose with straight chain should be cellulose, so option A. Number 9. Which row describes a triglyceride? Alright, triglycerides are hydrophobic. Yes, they are. Insoluble in alcohol? Well, they are soluble in alcohol, so it is going to be a cross. So option B it is. Which diagram shows where a peptide bond would be formed? Well, a peptide bond will be formed between CO and NH, and this water molecule will be lost. So I'll go with option A. Let's see why option B, C, D are not correct. B, well, this is the wrong position, and of course this is the wrong position too. This is the wrong position. I mean, this is even the wrong carbon. So yes, for question number 10, option A. Now let's move on to the next question. Okay, question number 11. Which statement about collagen molecules are correct? Number 1, both types of secondary structure occur. Number 1, both types of secondary structure occur within the molecules. Types of secondary structure. So we know that collagen molecule does not have this secondary structure. It neither have alpha helix. I mean, it has a special kind of alpha helix, and then it doesn't have a beta-bladed sheet. So collagen molecule, number 1 statement is wrong. Large number of hydrogen bonds stabilizes the molecule. Well, that it does. Repeated amino acid sequence says determine the tertiary structure. Glycine is repeated many times so that the protein can pack very nicely. So I'll go with option 2 and 3. Alright. The protein glutenin gives bread dough its elasticity. The diagram represents a polypeptide of glutenin, tightly coiled regions in 2 sides, and then loose spiral. What describes the structure of glutenin? Okay. Alright, let's see. Quaternary structure, because there are both globular and fibrous region. Okay, quaternary structure. Now as we can see, this is not a quaternary structure because there are not more than 2 proteins 2 polypeptides present, so we will just cross out A and B. Then C, secondary structure, because loose spiral is an alpha helix. Well, loose spiral is an alpha helix, I cannot confirm that. Alright. D, tertiary structure, because different regions forms a 3D shape. Yes, we can see the 3D shape here, so we can confirm that, and therefore I'll go with option D. Question number 13. Which diagram correctly shows hydrogen bonding between water molecules? The symbol partial indicates the partial charge on an atom in the water molecule. Alright, let's see. Option A has the hydrogen bonding, but then you can see there is a partial negative, so it's wrong. This is also wrong. So when it comes here, this is a wrong bonding in the first place. I'll just cross it out. And then we are left with option C, which is the safe bet. As you can see, partially positive hydrogen, partially negative oxygen. So yeah, this is the correct structure. So let's move on to question number 14. Aspirin inhibits an enzyme by reacting with an amino acid that forms an essential part of the 3D structure of the enzyme. Part of the aspirin molecule binds firmly with the amino acid. What describes this inhibition? Number 1, competitive inhibition. Okay, good question. All right. So you see, part of the aspirin molecule binds firmly with the amino acid. I mean, with the amino acid, whenever it is referring, then obviously it is not a competitive inhibitor, because competitive inhibitor would bind with the active site. So competitive inhibition, wrong. Reversible inhibition, wrong, because there isn't the other option left, which is the non-competitive inhibition only. So option D. The table shows Michaelis-Menten constant Km for 3 enzymes. Enzyme CPF. Okay. As we can see, even though the number minus is, the greater, but then this is the smaller value. Okay. Which interpretation of the information is correct? Enzyme C has a Vmax which is half that of enzyme P. Well, this information is insufficient to actually explain that, so I'll just simply give it across. Enzyme C will reach Vmax in the shortest time interval. Can't confirm that either. Enzyme F has the greatest affinity for its substrate. Yes, it does. And enzyme P has a Vmax of 6.0 into 10 to the power minus 3 millimole per dm cube. Enzyme P has a Vmax of 6.0. Well, cannot confirm that either, because Km necessarily doesn't give us the understanding of the Vmax, so we can't actually tell what is the Vmax or at what concentration Vmax is actually going to occur. So C is the correct option. Next question. The diagram is a drawing from an electron micrograph of a typical animal cell. Structure-wise, a mitochondria, I can say that. X, okay. How many layers of phospholipid will a molecule of oxygen cross to travel directly from X, okay, outside the cell to Y, the matrix of mitochondria? So there will be 2 here, 2 over here, and 2 over here. So I would say 6. Next question. Question number 17. How could water molecules cross the cell surface membrane of animal cells? Well, it can go through channel protein, and that's about it. Okay, because carrier protein carries things with energy, and cholesterol molecules are hydrophobic, so they won't even touch water. Question number 18. The diagram shows a xylem vessel in a root and 4 adjacent parenchyma cells. Okay, alright, good. As water moves up the xylem, it is replaced by the water from the cell D. Water in cell D is replaced by water in cell C, resulting in a flow of water from cell A to the xylem. Which cell has the least negative water potential? So the most negative water potential would be at the xylem. So the least negative water potential should be here at A. So I'll go for the option A. Agar cubes can be used to demonstrate the effect of changing surface area to volume ratio on diffusion. 3 different agar cubes made using a dilute acid were placed into an indicator solution that diffused into the cubes. When the indicator came into contact with the acid, it changed color. Okay, the cubes were 1 cm cube, 2 cm cube, 3 cm cube, and were left in the indicator solution for 10 minutes. All other variables were kept the same. The results were recorded as diagrams. The results for 2 cm cube are shown. So in a 2 cm cube, we can see that a depth of 0.5 centimeter has been penetrated, which reference represents the results for 1 cm cube and 3 cm cubes, not to scale. Okay, so now one thing I can say that it has to be a depth of 0.5 centimeter for both. Now since it is a 1 cm cube, so that therefore there is a 1 cm length in here, so 0.5 would be a complete. So according to my best of judgment, A would be the correct answer, since 0.5 cm cube from both sides would just simply cover up the entire structure. Okay, B also has a close call, but then this particular one is wrong because it has a depth of 1 centimeter, and which is wrong. In this side is correct, but this side is wrong because it has a depth of 0.25 centimeter. Cube change color. Okay, this is wrong, and this is wrong too. So yes, option A is the best choice. Number 20. The photomicrograph shows cells in different stages of mitosis. Okay. All right. We can see P in anaphase. We can see Q in probable telophase, and T in metaphase. R is in, looks like early prophase to me. S is in interphase. Which statements are correct? Cell T shows metaphase. Cell T in fact shows metaphase. DNA replication occurs in cell R. Nope, that is wrong. The amount of DNA in cell P is the same as in cell T. Cell P is same as in cell T. Yes. The correct order of the stages is S followed by R followed by T, P, and then Q. Yes, that looks correct to me. So option 1, 3, 4, I would go with option C. So question number 21. Which changes in a group of mammalian cells dividing by mitosis would be necessary for the formation of a tumor? Number 1, mitosis is no longer inhibited by cell-to-cell contact. Yes. Number 2, cells acquire the ability to migrate and set up new colonies. Not necessary. Number 3, cells become able to divide indefinitely. Yes. So option number 1 and 3. 21, B. Next question, question number 22. Which row shows some of the events during mitotic cell cycle in the correct time sequence? Centromeres of each chromosome splits. So this is anaphase. I would refer to as A. Nucleoli becomes visible. This is telophase. Nuclear envelope breaks down, prophase. Spindle microtubules are produced, prophase. So this is anaphase, telophase, prophase. Now it doesn't look like a correct time sequence. Nuclear envelope breaks into small vesicle. This is prophase. Spindle microtubules produced, again prophase. Centromeres of each centrosome splits. Okay, each chromosome splits. Okay, anaphase. Nucleoli becomes visible, telophase. Looks like a correct sequence from prophase to telophase. So I would go for this one. Let's look at the other options. Nucleoli becomes visible. Again, telophase. Nuclear envelope breaks down. This is prophase. Spindle microtubules are produced. This is not a correct sequence of mitotic cell cycle. Spindle microtubules produced. Spindle microtubules produced, centromeres. No, this is not A. So mitosis has to be PMAT. Prophase, metaphase, then anaphase, and followed by the telophase. And this one fits the criteria. So 22 option is B. I would say this was a little bit difficult question. What is the maximum number of hydrogen bonds in length of DNA containing 700 base pairs? Okay, for this particular one, so if it is 700 base pairs long, so each one of them can have like a maximum of 3 hydrogen bonds. Okay, that is between C with a G. So if we have like CG running 700 base pairs, we would have 2100 maximum number of hydrogen bonds. What is the common component of 3 molecules, RNA, DNA, and ATP? Well, common component, okay, of the 3 molecules. Now, the most common components of the 3 molecules, RNA, DNA, and ATP, I would say would be phosphate. Yes, adenosine is present in all of them. Hydrogen bond is not present in all of them. And ribose is not present in all of them. However, phosphate is the most common component. So, question number 25. Photomicrographs of a dicotyledonous plant root, stem, and leaf are shown. Okay. Alright. Now, let's name them beforehand. Would make life easier. Number 1 is xylem. Number 2 is phloem. Number 3 is endodermis. Okay. Number 4, I would go for number 4 is cortex, and number 5 is epidermis. So option number 1 is xylem. Now with xylem in option number 1 we have a D. So xylem, option number 2 phloem, endodermis, cortex, and epidermis. It's fine. Which features apply to phloem sieve tube element and to xylem vessel elements? Okay, phloem sieve tube elements and xylem vessel elements. No cytoplasm. Phloem sieve tube element contains cytoplasm. No end walls. Phloem sieve tube element contains end walls. No nucleus. Only option number 3. So the answer is D. 26D. 27. Which statement correctly describes an event in the process of mass flow in phloem sieve tube element? Option A. Any part of a plant where sucrose is loaded into a sieve tube element is called a sink. No, it's called a source. Loading sucrose into a sieve tube element increases. Loading sucrose into a sieve tube element increases the water potential inside it. It cannot. It will lower the water potential. Mass flow in the phloem is a passive process occurring at the same rate as diffusion. Cannot tell. Sucrose moves down a hydrostatic pressure gradient in the phloem from source to sink. This I would prefer as the correct statement. Mass flow in phloem is a passive process. We can't tell that because it occurs due to a pressure gradient. The diagram shows a potato tuber at different stages. Number 1, dormant, tuber not growing. Number 2, sprouting roots and shoots. Number 3, growing roots and shoots. Number 4, making new potato tubers. Which row identifies the stage where potato tuber acts as a source and a stage where it acts as a sink? Now number 4 is definitely sink. Okay, so source can be option number 2 and 3. So source, yeah, number 2, source is fine. So option number B, 28B. Question number 29. What describes the role of the atrioventricular node of the heart? Option A, it causes the muscles of the ventricles to contract from the apex upwards, not a function of AVN. It delays the transmission of wave of electrical activity from sinoatrial node. Yes, it delays. It initiates a new wave of electrical conduct activity in the ventricles. No. It provides a non-conductive barrier between atria and the ventricles. No. So option B, 29 is B. Number 30. The diagram shows pressure changes during the cardiac cycle. Which arrow indicates ventricular systole? Okay, this is very clear. Option B is the correct choice. Now, it is not option A because option A, I would say, indicates atrial systole. It is not option C because option C indicates ventricular systole, ventricular diastole, and atrial diastole both. It is not option D because that indicates shutdown of the semilunar valve. So option B it is. Number 30, option B. Which process can be carried out by a mature red blood cell? Option A, active transport. Option B, cell division. Obviously not cell division. Phagocytosis cannot do. Protein synthesis cannot be done as well. So active transport only. So option 31, A. 32. Hemoglobin can bind to carbon dioxide, carbon monoxide, and oxygen. Which statement about the binding site of hemoglobin is correct? Okay, so now you gotta understand this particular fact. Carbon monoxide and oxygen has the same binding site. So it is the carbon dioxide that has a different binding site. Okay, so which statement about the binding site of hemoglobin is correct? Carbon dioxide and carbon monoxide bind to one site and oxygen binds to a different site. Wrong. Carbon dioxide and oxygen binds to one side, carbon monoxide binds to a different side. Wrong. Carbon monoxide and oxygen binds to one side and carbon dioxide binds to a different side. Correct. Okay. Carbon monoxide and oxygen and carbon dioxide all different sides. Now that is also wrong. So option C, 32C. 33. Which tissues are present in a bronchus? So, alright. In a bronchus, cartilage will be present, ciliated epithelium will be present, and smooth muscle will also be present. So option A. This was a relatively easy one. Cigarette smoke contains tar. Which statement describes the effect of tar on the respiratory system? Number 1. Tar causes goblet cells to increase the secretion of mucus. Okay. It does. Tar increases the risk of blood clots forming inside blood vessel. Well, technically it's not inside the body. Tar may cause changes to DNA in epithelial cells. Yes, it can. Okay, so option number 1 and 3. So C. Symptoms of chronic obstructive pulmonary disease, COPD, include a cough, breathlessness, and chest infections. Some of the changes in the lungs that result from smoking are listed. Number 1. Alveoli lose elastin and collapses. Okay. Bacteria accumulate in the mucus. All right. Okay. Cilia are inactivated. Okay, cilia are inactivated. I think symptoms of chronic obstructive pulmonary disease, COPD, include a cough, breathlessness, and chest infection. Some of the changes in the lungs that result from smoking are listed. Number 1. Alveoli lose elastin and collapse. not the effect of okay, not the effect of smoking, as in it is an action of white blood cell. Bacteria accumulate in the mucus. Okay, again, so even bacteria accumulating in the mucus will not cause the COPD to cough in COPD. Okay, cilia are inactivated. So when cilia are inactivated, cough will be induced. Number 4, goblet cells secrete more mucus. Yes, more mucus means we need to move that mucus. Which changes causes the cough in COPD? So option number 3 and 4. I mean, COPD is caused by all the options, but then the cough is mainly caused by cilia are inactivated and goblet cells secrete more mucus. 36. Which statement about infectious diseases are correct? Number 1, cholera has a vaccine available and is caused by a bacterial pathogen of Vibrio cholera group. Correct. Measles has a vaccine available and is caused by a pathogen called morbillivirus. Smallpox was eradicated by vaccination and was caused by bacterial pathogen. No, it was caused by a virus. So option number 1 and 2. 36A. 37. The antibiotic tetracycline binds to small subunit of bacterial ribosome, stopping protein synthesis. Bacteria have become resistant to tetracycline due to the effect of mutations. Which effect could produce resistance to tetracycline? Okay, preventing tetracycline from binding to the bacterial cell wall. Okay, tetracycline doesn't bind with the bacterial cell wall. It binds with ribosome. So preventing tetracycline from entering the bacterial cell could be one of the issues. Preventing tetracycline from inhibiting transcription. Tetracycline was there to inhibit translation, so it's wrong. Preventing the production of tetracycline by ribosomes. Okay, wrong as well. Okay, so option B, 37B. Next, 38. Where are antigens found? On the surface of macrophages, yes, when they're acting as APCs. In blood plasma. Antigens can be found in blood plasma. Antigens are not found in blood plasma, so we can go for option B. 39. The graph shows how the body reacts following exposure to an antigen for the first and second time. Concentration of antibody, first exposure. Okay, increase, second exposure. What is the direct cause of the increase in antibody production after the second exposure to antigen? Yes, the plasma cells that are present in the blood at that time. Memory cells would be, memory cells will produce the plasma cells, but then it takes time. So if the plasma cells are present, they are the main cause of the increase in antibody production in the beginning. Last one. Why has it proved difficult to develop an effective vaccine against malaria? Mosquitoes have many stages in their life cycle. Human immune system does not recognize the antigen of the parasite. So both of these options are wrong, because mosquitoes have many stages in their life cycle. That has nothing to do with the vaccine. The human immune system does not recognize the antigens of parasite. No, it does. The parasite can only be attacked when outside the liver cells and red blood cells. That is the main reason. Vaccines are rapidly broken down by proteases in the stomach. Nope. Okay, so for question number 40, option C. Thank you everyone for joining the class today. Inshallah, we will be uploading more videos like this in the near future. Please leave a comment of how you would like the representation to be, and subscribe. Thank you very much.